Okay, so welcome back to this next video in which we are discussing adrenoceptors. Okay, right. So we now want to turn our attention to looking at each of the main classes of adrenoreceptor in turn and looking at their localization and what it is that they do, basically. Okay, so we're going to start off with the alpha-1 adrenoreceptor. Okay, so the alpha-1AR here. Right, okay, so we've seen that there are three separate types of alpha-1 adrenoreceptor. We've seen that there is the alpha-1A adrenoreceptor, there is the alpha-1B adrenoreceptor, and there is also the alpha-1D adrenoreceptor. We are going to consider them all together because their localization is pretty much the same, and what they do is the same. Okay, so where are all of these receptors located? Well, they're found in smooth muscle cells, both smooth muscle cells around blood vessels and also smooth muscle cells which surround other tubes, such as the bronchiolar smooth muscle, the smooth muscle surrounding the uterus, the smooth muscle surrounding the intestinal tract and places like that. Okay, so you have smooth muscle cells all over the body and they have a very important physiological function. Okay, and alpha-1 adrenoreceptors are found on the membrane of smooth muscle cells. Now what does it trigger? Basically, when you activate the alpha-1 adrenoreceptor with, for instance, noradrenaline or adrenaline, and remember, noradrenaline has a greater affinity for the alpha-1 adrenoreceptor than does adrenaline. Okay, when you stimulate alpha-1 adrenoreceptors on smooth muscle cells, what it triggers is the activation of the contraction of that smooth muscle cell. So the smooth muscle cell will contract when you stimulate the alpha-1 adrenoreceptor. And what we now want to look at is how that occurs, basically. We want to look at how stimulating the alpha-1 adrenoreceptor causes the contraction of the smooth muscle cell. Now, I think the best way to do this isn't actually to start with the alpha-1 adrenoreceptor, go through its pathway, and then look at smooth muscle cell contraction. I think actually it's better to look at smooth muscle cell contraction so that we've got that, and then what we'll see is how the alpha-1 adrenoreceptor is actually going to activate smooth muscle cell contraction. I think that's a more natural way to present this than to work through it in the logical sequence that it occurs. I think otherwise you end up going through the pathway that the alpha-1 adrenoreceptor activates, and then you have to go on a huge sort of divergent tour of smooth muscle cell contraction, and by the time you've done that, you've forgotten the way that the alpha-1 adrenoreceptor actually stimulates its pathways. So I think it's best to look at smooth muscle cell contraction first. I think it makes better logical flowing. Okay, so let's start by looking at the structure of a smooth muscle cell and then look at how smooth muscle cells contract. Okay, so smooth muscle cells have this sort of spindle shape, okay? So they have a big belly and then very thin ends, like so, okay? Now, their diameter, okay, at the thickest portion in the middle is around 1 to 5 micrometers. Okay, so remember, a micrometer is a meter divided by a million, basically, a millionth of a meter. Okay, so it's approximately a millionth of a meter to 5 millionths of a meter. Okay, meanwhile, the length of a smooth muscle cell from tip to tip is usually between 20 and then 500 micrometers. So depending on where the smooth muscle cell is, the length of it can vary quite largely. Okay, right. So we now want to look at how do smooth muscle cells actually contract, basically. How do they get smaller? Well, basically, if I draw another smooth muscle cell here, okay, inside of them they have a contractile meshwork. Okay, so they have these special bodies of proteins that you have sort of dotted around the cytoplasm of the cell. Okay, so we'll put one here, we'll put one here, we might put one over here, one here, and some of these will be attached to the membrane, by the way. Okay, so these are special aggregates of proteins, and they are called dense bodies. Okay, so some of these dense bodies are just within the cytoplasm, and some of them are actually attached to the membrane. So let me colour these in a specific colour. So I'll colour them in turquoise here. So here are the dense bodies which you have attached to the plasma membrane, and then you have some within the cytoplasm. And basically the idea is this, that between dense bodies you are going to suspend a structure which is capable of contracting. Okay, you're going to suspend what's called a contractile unit, and we will look in detail at the structure of these contractile units. But for now, just imagine putting something between these which is capable of contracting, and we'll just call it a contractile unit. Okay, and then you have them all over the place. You'll have another contractile unit here, you might have another contractile unit here, you might have a contractile unit here, a contractile unit here. Okay, so you have contractile units all over the place, forming a meshwork between these dense bodies. Okay, and I think I'll just put another dense body over here with a contractile unit there. Okay, so now can you imagine what's going to happen if all of these contractile units start contracting? Well, all of the dense bodies are going to be pulled closer to one another. Okay, so let me try and show this. Okay, and I realise that I am exaggerating it, but basically what will happen is all of these contractile units have now contracted, basically, so that they're all shorter, and what it ends up doing is contracting the entire cell, because the meshwork is all over the place, basically. So you pull the entire cell down, the whole thing contracts, basically, because of the contraction of these contractile units that is that are suspended between these dense bodies, basically. Okay, so what we now want to talk about is what is the actual structure of one of these contractile units. So let's put two dense bodies at either end. So we'll have one dense body here, okay, and then we'll have another dense body over here, and we now want to see what is this contractile unit that we suspend between the two. Okay, and I'll highlight the dense bodies again in turquoise, like so. Okay, right. So basically, attached to the dense bodies, what you will have is actin filaments. So from this dense body here, you'll have actin filaments coming out, and from this dense body here, you'll have actin filaments coming out. So I just want to discuss with you the structure of an actin filament. Okay, so let's discuss the structure of an actin filament. So basically, actin is a small globular protein, and when it's just on its own as a little monomer, it's specifically called G-actin. If you want to stress that it's the globular monomer, it's called G-actin. So this is a little globular protein, which I'll just draw like so. So this is globular actin here, or G-actin. However, G-actin can polymerize up into huge great filaments, and the filamentous form of actin is called F-actin. Okay, so this is a polymer of loads of these together. And how does it polymerize together? Well, basically, it polymerizes together to form an alpha helix. So basically, it will wrap around and around and around like so, and it will form basically a little alpha helix. Okay, so it's like a spring structure. So I'll draw it like so. So that's the sort of shape it's in. It's in a sort of spring shape, and the fancy word for a spring shape is to call it an alpha helix. Okay, so we've got loads and loads of these G-actin monomers polymerized together to make an F-actin molecule, which is this filamentous form of actin. Okay, so I'll colour these all in pink to show that they're the same protein as the G-actin, just polymerized together. And basically, if you then want to build an actin filament, what we're going to do is take two F-actin molecules, and we're going to create a double helix of them. So if I draw this single alpha helix out again, I'm going to then wrap another alpha helix around there to make a double helix. Okay? similar to DNA, a single strand of DNA is an alpha helix, but when you put the double strand in, it then becomes a double helix. So here comes the other strand that will wrap around like this, and it becomes quite difficult to show because they're overlapping one another. Like so. Okay, so you put this other strand of F-actin around it to make a double helix. So if I colour this in, hopefully it'll look nicer. Right, so all of these are still G-actin monomers polymerized together into a great big strand like this. Okay, and this sort of a structure is called a double helix, and it's just like DNA basically. It's got two alpha helices wrapped around each other like so, and this is what we're going to use as actin filaments. This is what we mean by an actin filament. Okay, so this is a double helix, or we could call this an actin filament. Okay, so it is actin filaments that we are going to attach onto these dense bodies, and from now on, what I'm going to do is I'm not going to draw them as alpha helices because it will take, double helices because it will take too long. I'm just going to reduce it down. I'm going to demote it to being represented just by a line like this. Okay, and I will colour it in red since I've used red continuously for actin. Okay, so basically, coming off these dense bodies, you will have large amounts of this double helical actin. Okay, so here are these actin filaments coming off. Now, I only draw 3, but in reality, you'll have hundreds of the things coming off here. And this one will also have actin filaments coming off it. So basically, each of the dense bodies, each of the 2 dense bodies that are going to be involved in this contractile unit between the 2, they have these double helices of actin molecules that are sort of pointing towards each other. So this one has actin filaments pointing towards this one, and this one has actin filaments pointing back towards this one. Okay, so these are the actin filaments. And as I say, even though I've only drawn 3, that's an under-exaggeration. That's to make the picture look nice and easy and clear. In reality, you'll have hundreds of these things. Now, how long generally are these actin filaments? Well, generally they're around 1 micrometer in length. Okay, so they're very small little filaments. Okay, right. Now, you're also going to have another important filament that's going to make up the gap, because as you can see, these actin filaments don't reach each other. There's a gap in between them, and basically you're going to stick another filament that will be in between them and which will overlap the ends of the actin filaments coming from both of the dense bodies. Okay, and this other type of filament is what's known as a myosin filament. Okay, so this is going to be a myosin filament, and I think I'll colour in the myosin filament in blue. Okay, so what I now want to discuss with you is the structure of a myosin filament. So we'll start off with the structure of a myosin monomer, because again, myosin is a polymer of many different subunits stuck together. Okay, so let me just grab another piece of paper. Right, okay, so let's discuss the structure of a myosin monomer then. Okay, right, so the structure of a myosin monomer, this is a little bit more complicated because depending on who you go to, you'll get a different answer. Okay, now officially, this is what a myosin monomer is. So I'm going to draw you officially what a myosin monomer is. But then when we're actually discussing how myosin filaments work, it will be easier to think of the myosin monomer being something different. Okay, and I'll explain all of that in a moment. Right, so what I'm about to draw is officially what a myosin monomer is. Okay, so basically you have 2 myosin heavy chains. Okay, and these are also alpha helical proteins. Now, each of the myosin heavy chains is just a single protein, but it's an alpha helix like so, and then it will go off in this sort of direction like so. And it might have a little bit here. And then you will have another myosin heavy chain here that will make it into a double helix, like so. And then it will go off in this direction and then form its head over here. Right, so let's colour these in separate colours so that we can easily distinguish the two. So, this is a myosin heavy chain here in red, and it's one protein, okay? So here is one protein, and it's got an alpha helix like so, and then it's got this portion up here which is going to be called the head, okay? Then you've got another myosin heavy chain here in turquoise, which also has an alpha helix, which is sort of wrapping around the alpha helix of the first myosin heavy chain, and that forms a double helix, and then it's got a head coming off up here as well. Okay, like so. Right, so we've got these two myosin heavy chains, so I'll just label them up. So this is what's known as the myosin heavy chain, and we've got two of them in a myosin monomer. Okay, so myosin heavy chain. And myosin heavy chain is often abbreviated, rather confusingly, to MHC. Now, generally, if you hear MHC, you do not instantly think myosin heavy chain. Instead, you'll probably think major histocompatibility complex. Okay, so be aware that use the abbreviation MHC at your peril. Okay, make sure it's obvious which MHC you're talking about, because people will not, when they hear myosin heavy chain, when they hear MHC, think myosin heavy chain. They'll think you're talking about something in immunology. Okay, right. Then this isn't the end of myosin. You also have, associated with each of these heads of the myosin heavy chains, you have two myosin light chain proteins, which I'm just going to show like this. Okay, now one of these is what's known as the essential myosin light chain. Okay, so we'll call this one the essential myosin light chain, and the other is what's known as the regulatory myosin light chain. So this is the essential myosin light chain. Okay, and myosin light chain is usually abbreviated to MLC for short. Myosin light chain, and you can do that more safely than you can abbreviate myosin heavy chain to MHC. And the other myosin light chain is known as the regulatory myosin light chain. Okay, so this is the regulatory myosin light chain, or the regulatory MLC. Okay, right. So let me colour in the regulatory and the essential myosin light chains in different colours. So we'll have the essential myosin light chains in blue, and each of the heads has an essential myosin light chain. Okay, and then we'll have in orange, if I can find orange, here we go, the regulatory myosin light chain in orange here. Okay, so this is the actual structure of a myosin monomer. So it consists of two myosin heavy chains with two myosin light chains associated with each myosin heavy chain. So overall we have 4 myosin light chains. Okay, so this is a myosin monomer officially. However, basically this myosin monomer really consists of 2 separate functional units. Okay, so each myosin head with, each myosin heavy chain with 2 myosin light chains associated with it is a functional unit on its own. So often people will just think of a myosin monomer as being 1 myosin heavy chain with 2 myosin light chains. But officially, well, really, you do have them associated in these dimers like so. So, this portion down here is what's known as the tail, okay? And each of these portions here, so this portion like so, this is then known as the head. So, each myosin monomer has 2 heads, basically, okay? And this portion that sort of leads from the tail to the head, this is known as the arm, okay? Which is why people often draw pictures like so. Okay, so they'll often draw a picture like this for myosin. And as you can see, this really only takes account of half of a myosin molecule. Okay, so this here is the tail, but this shows you 1 tail. So let's say we are looking at this red myosin heavy chain here. So this is the tail, then we've got the arm here, and then we've got the head here. So people often might draw pictures of myosin monomers like this, and really what they're showing there is half of a myosin monomer. Okay, they're showing the functional unit, because basically a myosin monomer actually contains two of these, and both of them function separately. Although they are beautifully attached via this double helix, the two heads are going to function separately, basically. Okay, right, so that's the sort of dispute over the real nature of a myosin monomer and then the functional nature of a myosin monomer. So don't let that confuse you. Okay, right, so let's now discuss the structure of a myosin filament then. So basically a myosin filament consists of around 200 myosin monomers. Okay, and when I say 200 myosin monomers, I mean real myosin monomers, so these things. So 400 if you're thinking about the functional myosin monomers. Okay, and you can basically split the myosin filament into two halves. Okay, we can split it down the middle, and basically this is because half of the myosin filament is going to interact with the actin filaments over here, and the other half is going to interact with the myosin filaments over here. Now, what is this contractile unit actually trying to do? It's trying to pull these dense bodies towards one another. Basically, it is the myosin filaments that are suspended in the middle that are going to do this pulling. Now, how are they going to pull? Well, this myosin filament here is going to pull on these actin filaments here, and they're going to pull them in this direction, okay? Whereas the half of the myosin filaments that are on this side are going to act on these actin filaments, and they need to pull them in the opposite direction, okay? So in fact the machinery needs to be the opposite way round on this side, and we'll see the importance of that in a moment, okay? So that's why I will divide the myosin filament up into two halves. Okay, so basically, a myosin filament is around 1.6 micrometers in length. Okay, so they're longer, slightly longer than the actin filaments, okay, which were 1 micrometer. And basically, you stick loads of these myosin monomers together to make one of these filaments, and all of the tails end up stuck in this sort of body of the myosin filament, as it's called. So this is called the body of the myosin filament. And then sticking off the sides, you have the arms, okay, of the myosin filament, of the myosin monomers. And then you also have the heads of the myosin monomers, and people generally are reduced to using this picture to denote this. So we will denote each of the functional monomers, if you like, although that's not common terminology. We'll denote each of the functional monomers separately, basically, rather than acknowledging that they're really as these dimers, like so. Okay, and basically, as you can see from my picture, the arm is tilted in a certain direction. Okay, you will have the arms that are on this side, which are interacting with myosin filaments, actin filaments over here, and want to pull the actin filament in this direction here. You will have them tilted in this direction. Whereas on this side, where you have an actin filament over here, and you want to pull it in this direction, you will have the arms tilted in this direction. So on this half, all of the arms will be tilted in this direction, like so. Okay, and you'll have arms coming off all over the place, basically. It's not just on the top and on the bottom, they'll come off on the sides, all over the place. But all of the arms will be oriented, pointing in that direction, basically. And on this side, all of the arms will be oriented, pointing in this direction. Now, this means that there is a little portion in the center where there are actually no heads and no arms coming off the myosin body. Okay, and this goes on for about 0.2 micrometers. And you might ask, well, why is this? Well, if you think about these little units here, basically, what's going to happen is we're going to have it like so. Okay? And we're going to have these ones oriented like this, which means that there's a region where you just have tails in the center, and this is this 0.2 micrometers in the center, basically. Okay, where you don't have any heads or any arms coming off, and it's because this is the point where the orientation of them has to change over, i.e. they have to move around 180 degrees. Okay, so you have a portion where there are no heads and no arms coming off. Now, I'd just like to give you a bit of terminology. The name for a myosin arm and a myosin head, okay, combined together. So this portion of a myosin functional monomer, if we like, okay, this portion is called the myosin crossbridge. Okay, so this is called a myosin crossbridge. Okay, so what we can say is that there are no myosin crossbridges coming off from this 0.2 micrometers in the center of the myosin filament, basically. But on the two other halves, you do have myosin crossbridges coming out, and they're oriented in the opposite directions. Okay, right. So we're now ready to discuss the sliding filament mechanism of muscle contraction. Okay, but I think we'll call it there for this video, and we'll do that in the next video.